So I'm really honored to introduce our next speaker. So Dr. Pierluigi Nicotera is the director of the German Center for Neurodegenerative Disease, the DZNE. This is a collaboration that we're forming with the University of Wisconsin, so we're thrilled to have him as a leader of that program. He has been a world leader in neuronal cell death related to neurodegeneration, and so we look forward to his talk on translational research in Alzheimer's disease. So we'll move. Okay, so are you going to switch? Okay, thank you.
Well, first of all, Cynthia, thank you very much. Sanjay, thank you for inviting me here. It's a great pleasure to be back again here in Madison. And I also would like to say that this is really a great collaboration we have started. And the interests are joining from different sides now as we go and we progress towards more practical agreement and research. We see that there are a number of points that we are really working on together with the same kind of expectation and perspective for the future, in particular on the role of aging and age-related diseases. And I will touch a little bit upon this. I will try to sort of give you an overview of the different or new ways by which we at DZNE are trying to sort of find treatments for neurodegenerative diseases in general, with the main emphasis on Alzheimer's disease. Obviously, the main problem that we have in neurodegenerative diseases is, particularly in Alzheimer's disease, is that we have no measure disease-modifying therapies. It is amazing that sometimes I have to speak also comparing neurodegeneration with other diseases. And while the problem in cancer, it is that the FDA, for example, has licensed so many medicines which have actually small benefits for cancer patients because of personalized medicine. We see improvement like, in 2 months average lifespan in many cancers. We have yet not too many neurodegenerative diseases. So as personalized medicine and stratified medicine goes on, also in neurodegenerative diseases, we hope that there are going to be accelerated pathways from the FDA and from other agencies in the world to be able to approve medicines for neurodegeneration. The main problem is that the causes are still unclear in many cases. Are we talking about syndromes? I mean, dementia is in a way a symptom, and it may be brought about by a number of different causes. I mean, we know about vascular dementia, we know about mixed forms, which are perhaps the majority. We know Alzheimer's disease, we know frontotemporal dementia, and so on. We will discover a number of conditions that are actually different one from the other. So one mistake has been to assume that necessarily the mechanisms are exactly the same every time we face a dementia. The other problem is that the factors that link aging to neurodegeneration are still unclear, and I will touch upon some of those, but this is a key issue. The main risk factor for Alzheimer's disease is age, old age, and we still have to clarify what factors are involved. The other problem is, as I say, disease stratification, so younger, older individuals. Are we extrapolating too easily from the early onset Alzheimer's disease to the disease that we observe in individuals who are above 85, 90? What are the factors which make some individuals resilient to get this disease? It's amazing that the risk factor drops after the age of 95. Well, you would say, well, most people die. Well, they don't. We're getting older and older, and yet if you look at the risk, it drops after the age of 95, which means there are some resilience factors there which we need to understand. Of course, we heard from Eddie just before that the disease-relevant animal models are very few, and I was quite intrigued by the results that he presented today. You may have seen an article also in Nature Communications not long ago from an Italian group showing that actually they believe that Alzheimer's disease begins in the ventral tegmental area. It doesn't begin in the hippocampus. It doesn't begin in the hippocampus. It is something which begins in areas below. And they, of course, this may very well be true for conditions in which depression is one of the early symptoms, but it's actually an intriguing finding. And so probably shall we sort of, go back to the humans first and try to understand better what is the clinical development in early stages. And of course, we have the lack of strong predictive biomarkers. I think this is a search which is ongoing both here in Wisconsin, in many laboratories in the world, at DZNE as well. And of course, the clinical trials have a weak design, because the problem is that we rely on cognition methods which are not sophisticated enough and strong enough to be able to tell us whether there is a real improvement. We need more better ways to assess improvement in cognition, but also clinical trials, because of the regulatory agencies' requirements, are based on the improvement in cognition. Effectively, we should try to make the trials exactly for what the medicine should work for example, aggression. For example, changes in the behavior of the patient. There could be cholinergic drugs, for example, which would be working very well on aggression and some of the other symptoms of Alzheimer's disease without necessarily affecting cognition whatsoever. So I think this is another important message. And of course, I mean, we don't have yet models for large data integrations, which is something that we're trying to pursue in different ways. So how can we put data together from completely different models, from completely different laboratory finding? So this slide is familiar to all of those who work on Alzheimer's disease and illustrates in principle the fact that the disease begins way, way before then the symptoms appears. So the molecular determinants of Alzheimer's disease are actually pretty much showing up at the very early stage of life. And in a way, I mean, if one would be to be provocative, one could say that the early onset Alzheimer's disease, the genetic one, which is a small percentage of all cases, probably is a disease which begins directly before birth. And actually, we will then have it manifested when individuals are coming to their 40s and so on. It could be something which is so strongly inherited in that respect that the traits are present in young individuals who are, for example, 20 years old when their parents get the disease at 40. So this is the conclusion, one of the conclusions of the Diane study, that some of the alterations in Alzheimer's disease in children from people who have the disease manifest about 20 years or more earlier than the expected onset of the disease. On the other hand, is actually the same thing taking place in individuals who get the disease later. So of course we need different type of cohorts. We need the healthy mutation carriers, who are the children of people who get autosomal dominant Alzheimer's disease. We need risk cohorts, people, for example, with ApoE4 homozygosity and other type of risk factors. We need the population studies to look at the risk effect of through life, and particularly the role of aging and aging-related events for the risk. And of course, patients cohort in the end. And clearly, as Ed has just been saying, we have been focusing on the A-beta tau hypothesis for quite a long time. Do we need new hypotheses, or do we still need to be able to work on these two hypotheses, which is the key question in the field? I am convinced personally that we shouldn't let these hypotheses go on, because it explains a number of things that happen in Alzheimer's disease. But on the other hand, we should try to figure out what is the role of these pathological changes that we observe in the development of the disease, whether we can actually pinpoint or what causes these pathological changes in individuals who have no mutations, and at the same time to try to find therapeutic interventions which differ from those which have been done so far, in particular the antibodies against A-beta that are administered to people either with mild cognitive impairment or with overt disease when it is too late, and therefore they may not have any efficacy there. Or, for example, if we could actually try to do combined therapies which would address individuals with early diagnosis that may not have yet a fully overt disease, they may be more effective, lowering also the risk of side effects. The tau therapy is actually, there are trials ongoing at the moment, and at least we don't know yet what the outcome is, but the indications seem actually quite positive. I talked to Dave Holtzman, who has been one of the pioneers in this field not long ago, and he said he's very hopeful that the antibodies against tau may actually be functioning better than those against A-beta. Of course, we will see. what happened, clearly the tau antibody may target a wider number of diseases than Alzheimer's disease itself. So how can we improve the therapies, and what is combination therapy? So one thing we are doing together with Stefan Lichtenthaler in our DZNE unit in Munich and Christian Haas, we're trying to, for example, try combined therapy that will affect BACE1 and the beta secretase process in combination with the antibodies. Now, you know that BACE therapies have actually been not very successful. The recent one has not shown very much of effect. There are also side effects of BACE antagonists, but the combination therapy may actually be quite promising. And to do this, I think what Stefan is doing has developed a new proteomics method to be able to check using click sugars as a bait, both proteins which are substrate from BACE into the serum, into the CSF. So the result of sheddase or shedding of proteins, in this case BACE is a sheddase, and also to be able to identify cellular substrates. In this way we can understand what are the proteins which we should be very careful to block shedding of, which means the side effects that we may have from BACE therapy. And based on this, actually now together with Roche, they have developed some molecules which may be very, very useful in combination with the antibody therapy. So this is definitely one possibility, to be able to look into the same pathways, but with a renewed eye for combinational therapy. The other thing that has been alluded to by Eddie before is the transmission or the spreading of disease. You may all be aware of the controversy whether or not these diseases are prion-like versus not prion-like. I personally do not like the term at all because there is no evidence, in my view, that these diseases are transmissible in any form. Prion-like, meaning templating and spreading of disease, has been advocated by many, including by Matthias Jucker in the ZNE, but also in the ZNE we had our group, like for example Dino Di Monte, who actually clearly show for some of these proteins they do not behave as prion protein, but they do spread. So in this experiment, what Dino decided to utilize a classical model system, the overexpression of alpha-synuclein. You know that people get Parkinson's disease with triplication of alpha-synuclein. So he injected into the vagus a virus which would express alpha-synuclein. The virus, of course, goes retrogradely and travels to the brain and expresses alpha-synuclein in the brain. And he looked at the spreading in wild-type animals as opposed to knockout animals for alpha-synuclein. Now, you know that the prion hypothesis is based on the fact that you need a templating. So, in other words, if you treat a knockout animal for the prion protein with prions, you will never get the disease, or you will never get spreading, you will never get any pathology. Well, in this case, it's exactly the opposite. So animals which were injected and they were knockout animals have a much stronger spreading of this alpha-synuclein through different neuronal system, which is depending on activity of the neurons. So probably, as Ed was saying, on presynaptic activation. And at the same time, these animals then have the spreading of the alpha-synuclein accumulation throughout the brain much better than the animals that are wild type. So exactly the opposite of what you would expect based on the prion hypothesis. Actually, they went further now, and they have data in which they show that propagation of alpha-synuclein may also happen from the brain into the gut. So in a retrograde manner, going down to the vagus and then infecting the gut, or infecting or spreading to the gut, where of course it can give some other pathological features, and as it has been discovered initially by the Brack theory that, you know, part of the alpha-synuclein you can find in the enteric plexus. Let's go to another approach. So, you know, targeting spreading may be important, but maybe spreading is actually a result of synaptic activity, as we heard before, both pre- and post-synaptic activity. So that would be one way of interfering with disease. But what about the immune system? The immune system is becoming more and more important because it actually mediates some of the effects that we see in some of the transgenic animal models. And because there is evidence clinically that patients with Alzheimer's disease usually get worse after they have a systemic inflammation or episodes of systemic inflammation. So in this model system that has been developed by Michael Henneke and Eike Latz in Bonn at the ZNE, they discovered a couple of years ago that if you take an animal that is knocked out for the NALP3 inflammasome, which is a component of the innate immune system, well, these animals actually do not develop the alterations in the water maze that you would expect based on the classical APP-PS1 transgenic animal model. So in other words, these animals have a behavioral effect, which is impairment into the water maze test, but if you cross them with the NALP3 knockout mice, you see that basically they recover the alterations. So the whatever the water maze means, we can discuss here now for half an hour of the implication of the water maze, but obviously as a sort of a memory and learning test, it is recovered. It's interesting also that in these animals there is less amyloid deposition in animal models, which are usually very prone to accumulate amyloid. I'm very careful as defining these as Alzheimer's model. These are models of amyloid deposition. I think we heard this just before, and I would like to reiterate this. So in this model, at least you have a decreased deposition of amyloid, but at the same time, you have also an improvement in behavioral conditions. So what is the mechanism for this? And this is actually, oops, I'm moving the wrong thing. And this is actually not published yet, is in revision now in Nature. And the mechanism is actually quite surprising. So apparently what happens is that if you take the NALP3, which is a component of the inflammasome with these three main regions, these regions need another protein called ASC, which through the binding through the pidosome, the PID, actually make these proteins accessible to activate caspase-1. It is a sort of a docking protein. You can see here the CAR domains coming together, caspase domain of caspase-1. This is the classical mechanism of activation of caspases through the inflammasome. Actually, what Michael discovered and his collaborators, it is that ASC itself, when it is formed and docked, it is thrown out by the neurons, and it is essential to be able to get A-beta oligomers forming plaques and producing A-beta propagation. So in other words, if you knock out ASC, you are not going to have A-beta formation, plaque formation, but at the same time you're not going to have any propagation according to the classical model in which you seed A-beta in the brain. So this actually tells us that in the mouse model at least, you need the innate immune system to form A-beta. Without the innate immune system, you're not going to have A-beta formation. So this may be an interesting target because obviously, I mean, you may just want to remove all the A-beta that you can, but at the same time, you will still produce oligomers. Now, what if you would use, for example, ASC antibodies? You do not want to inhibit the inflammasome altogether, otherwise we would basically lose antiviral defenses. But you may just go downstream, and that's one pathway we are pursuing now. We have identified two kinases, which are essential in this pathway, and we try to inhibit that and see whether we can actually produce no side effects or low side effects, but at the same time block formation of plaques and propagation. Another component of the innate immune system which has received a lot of attention is TREM2. TREM2 actually missense mutation reduce the transport of this molecule on the cell surface, its shedding, and also the phagocytosis ability of microglia to remove A-beta. And Christian Haas and his collaborators have actually demonstrated this very nicely, and they have demonstrated that in disease, in marked disease, the CSF levels of TREM2 actually decrease pretty much. So which indicates that the ability of this molecule to be transported, to be functional at the surface of microglia is very important during disease. It goes down, meaning that it doesn't function, it doesn't do what it should normally do, which is probably facilitate phagocytosis of A-beta by microglia. The interesting thing is that when does this defect occur? It is a defect which is upstream or downstream to the classical mechanisms of Alzheimer's disease. And in a more recent study done in collaboration with the Diane Network with a number of patients who are carriers but not sick individuals, it turns out that actually TREM2 goes up in the CSF of individuals who are not symptomatic, so 7 years before the appearance of the disease. So it seems like it behaves exactly like the beta and tau ratio. So at the beginning it goes up, probably because there is an enhanced activity of TREM2 to be able to remove and to be able to scavenge amyloid which is accumulated in these individuals. But then as the disease progresses, it will go down and you will find less in the CSF. It is interesting that these changes occurs after the initial changes in a beta and tau ratio in the CSF. So it seems to be downstream to what the beta tau ratio will signify. So this is another interesting observation which implies that we could use this as a marker, the biphasic change in TREM2, but also that there could be a very important role in trying to restore the ability of microglia in scavenging these proteins like beta and tau. Let's move to another example, which is coming from our group in Berlin, from the entire site now, which is focusing on this. And I'm sure all the clinicians know that there are a number of antibodies which are found in the CSF and in the brain against brain proteins like ion channels, for example. And this is the case for a number of diseases. What we actually find in the brain, that in many cases, they're not only in encephalitis, but also in Alzheimer's disease, in ALS, FTLD, and Parkinson's disease, there are anti-NMDA antibodies. These are common in different forms of dementia, and now we have patented a system by which these antibodies can be removed. It's an apheresis system. Most of the people with encephalitis are going to plasmapheresis, and of course you remove all the antibodies. But if you have a selective system to remove only the bad ones, then of course you can try to keep the positive effect on a number of other serum factors. And we are testing this now to be able to see whether the potential implication of these antibodies is a realistic thing also in Alzheimer's disease, because the idea is there may be side effects or you know, a side whatever effect of the degenerative diseases that you have these antibodies there. But at the same time, I mean, this looks very promising because we do find correlation between different symptoms in patient and recovery. In the encephalitis patients, you actually do recover cognition when you use these antibodies. So there is a hope that, given the fact that, especially what Ed was saying before, that this potential synaptic recovery, if there is a mechanism that involves autoimmunity, in this case we would be able to neutralize it. Again, let's move. It's a panorama of finding, and I'll tell you, and I'm coming towards, and then, you know, the part which is more related to aging. I mean, another thing which we're trying to do is to be able to look at brain regeneration. You see this piece of spinal cord here. It is a white piece. It's still present here, but it has been cleared. So the technology of brain clearing is actually being developed by Frank Bradke and his collaborator in a paper in Nature Medicine in 2011, and then has been used in different laboratories around the world to clear the brain. So in other words, to be able to have a tool whereby you can use microscopy of fluorescently labeled neurons to be able to look in depth into the brain structure. And in this case, for example, you see the spinal cord. You can actually image the entire spinal cord, and you can go directly into the detail of these fibers. You can actually be able to image and see fiber by fiber. And this tool has enabled us to be able to study the effect of different drugs on spinal cord injury, which is, of course, a different form of degeneration than the one we observe in brain, but we're trying to extend this now also to conditions like Alzheimer's or Parkinson disease and degeneration. And using this technology, Frank and his co-workers have been able to show that if you use epithylon, which is a Bristol-Myers Squibb drug, you can actually help neurons in green to grow back through a lesion in the rat. And there is a functional recovery actually in these animals after the therapy. So we're trying to use this technology. So I gave you a little bit of sort of, you know, example of different kind of things that may be used in the future in therapy, or the ways at least we are pursuing different strategies beside the classical beta tau therapy. Now let me switch gear for the last 10 minutes and tell you a little bit about epigenetics. And 2 minutes? My computer say, my computer says 23 minutes. So here we have a divergence in the clock. All right, okay, I'll try to wrap it up quickly. Sure, no problem. So, of course, I mean, you've heard about epigenomic. I mean, there's one concept which I would like to sort of bring back. The idea is that we live longer, that's clear. If you look at people with, for example, Down syndrome, they actually double their lifespan today. Of course, we can cure them back. The cardiovascular responses are better taken care of, which means that basically they live longer, but they also have to adapt to this longer life. Exactly like us. We were living a century and a half ago, 50 years in average, and now we live nearly double. This is not only, of course, this hygienic condition, this medical treatment and everything, but how do we adapt? A 60-year-old man now performs much better than 60-year-old man performing a century ago, which means you need an adaptation. This adaptation cannot be a simple adaptation of a genomic response. It cannot be, because of course there is too short time for the genome to adapt. So it must be definitely an epigenomic adaptation. And if it is an epigenomic adaptation, then we need to be able to understand what is the mechanism or the mechanism that will help us to adapt to these kind of changes. Epigenomic adaptation does not mean something which is different from one individual to the next. It can be inherited. In this experiment, Dan Henninger in Bonn has mated old mice with young ones and look at the offspring of old father versus the offspring of young fathers, and turns out that the old father's offspring is much more sensitive to disease and has learning and memory disabilities. So we can inherit this kind of epigenomic changes. On the other hand, we have our own history, the genetic history, and this nice paper from Chris Walsh showed how well neurons are different. There are very, very few neurons which actually have completely different genome because of mutations acquired not in a replicative way, but directly during transcription, during aging. So, you know, what is basically the role of these mutations which we carry and the epigenomic changes and how they're linked together? Well, of course, one possibility to explain this is epigenetic changes together with accumulation of damage may actually make certain individuals more susceptible. It's an obvious hypothesis that we have there. And I'll skip this because basically it just tells you that one of the changes could be, for example, telomere changes. And I will go primarily on one particular topic, which is the histone H3.3, which is one of the ways by which you can change epigenomic structure. So you don't need to change methylation or acetylation. It is sufficient you change the ratio at which one histone, the H3.3, accumulates into the chromatin. Histone H3.3 is specific because it accumulates continuously during life. In non-mitotic cells, the other histones cannot be turned around. So then what happens is the only one which accumulates is this, and if you basically have an aberrant accumulation, you have genomic instability, aberrant gene regulation, and altered chromatin structure. And this is actually shown very nicely in this paper by David Hallis and his cooperators, that during time in brain you accumulate H3.3, and if you have a right turnover you have better learning and memory, but if you accumulate it then you impair learning and memory. And I'll show you just a few data that we published very recently. We decided to investigate what actually H3.3 would do to the aging process itself, and together with Daniele Bano in my group we actually generated a worm, a C. elegans, which is knocked out for the two homologs of the H3.3. And if you normally look at the lifespan of the worm, which is this black line, if you block the insulin signaling pathway, the IGF signaling pathway, you have an extended lifespan. If you knock down the two isoform of H3.3, you change nearly 40–45% of the genome. In this volcano plot you can see here, you change genes which are either upregulated or downregulated. Interestingly enough, nothing happened to the lifespan of the worm per se, but what you do, you completely abolish the extended lifespan that you get by blocking the insulin pathway. So which means you block completely the possibility of responding to the lifespan extension in the worm, which is also generated by stress or by other signals. I'll skip this, which basically is another evidence from our lab showing again that this can happen by normal neuronal stimulation. And go back to the last slide, which I think is relevant to show, which is metabolism. And this is a beautiful work from Jens Bruning, for which myself and Michael Hennecke wrote a news and views, which indicates you another important target of aging, which is metabolism. And what they show it is that if you take an animal and you give them a high-fat diet, this animal will turn down the glucose transporter into the brain, and therefore the brain will not have glucose. So what happened, the animal triggers a peripheral inflammation, mobilizing macrophages. They go to the blood-brain barrier, they start to reactivate the GLUT1 transporter via VEGF, and finally the brain can take glucose away. They can use glucose away. What is the consequence of this? Well, in the first place, if you block glucose transport, you block down the possibility of forming plaques in the brain. So if you reactivate this, then you form more plaques in the brain. The second consequence of this, it is that if you reactivate glucose transport, the brain triggers diabetes. So in other words, the brain, in a very selfish way, decides to be able to retain its glucose consumption at the expenses of what the periphery can do. So this is basically a concluding remark. I will skip the last slide is just to see how we do drug development. We have a cooperation here with the University of Wisconsin and a strategic cooperation with Orion Pharma, which has been developed here in Wisconsin as well in cancer research. And I think this is a different way by which we can actually explore all these new mechanisms and these new things together. And the last slide is the building. Actually, this is an old picture. Now it's all beautifully and done and finished. And we just opened it recently with the Disney structure, the people who have contributed to this work, and I apologize if I've been a bit longer. My computer says exactly 30 minutes. Thank you.